Welcome to the online lecture this week for ANP2. Because of the snow issue we've had the last couple of weeks, we'll be doing this week online in a virtual class. What will happen is this PowerPoint presentation will be presented with some commentary and some visuals. Along the way, there'll be some questions and some activities to complete. And then, as usual, when you're finished, you can go online and fill out your weekly worksheet and quiz. We'll be in Chapter 16, which is the lymphatic system this week, so let's get started. One of the main functions of the lymphatic system, as we see from this slide, is to move fluid throughout our tissues and back into our cardiovascular system. As we found out last week, a lot of that fluid becomes interstitial fluid and filling in the spaces between cells. The lymphatic system's job is to transport that fluid that accumulates in those spaces back into the bloodstream here. In addition, we see two supplemental functions: transporting fats to the bloodstream and helping to defend the body against diseases using our some white blood cells. From last week's lesson, we saw that a small portion of the plasma that escapes the capillary networks becomes interstitial fluid, and one of the primary responses of the lymphatic pathways is to move that fluid back into circulation again. So you see we have two networks of lymphatic capillaries that will pick up that interstitial fluid, traveling through something what we call a lymph node, which simply houses some of these white blood cells, particularly B lymphocytes, and monitoring that fluid for any pathogens present as it makes its way back into the subclavian vein. Here we see a comparison of some of those lymphatic capillaries along with the lymphatic vessels. As you can see, the lymphatic capillaries have a terminus end to them, whereas capillary networks really are a continuous circuit. The pressure is fairly low in these lymphatic capillaries, and they're surrounding the interstitial spaces of most tissues. From the last slide, we mentioned that the pressure is very low in some of these vessels due to the lack of blood pressure. The walls are and the structure of some of these lymphatic capillaries are actually very similar to blood vessels with regard to valves. As you can see here in the illustration, they're showing you this flap of tissue. It's just a flap of the endothelial lining of that tissue. Because the pressure is so low, there really is no hydrostatic pressure moving fluid through these vessels, so it really relies on body movements and other factors to encourage positive flow of lymph, which is the name of the fluid, through these vessels. So we've established that one of the main functions of the lymphatic system is to move this lymph fluid that escapes our capillary networks and drain that lymph into a series of lymphatic vessels. Obviously, as you can see here from the name, that they're derived from their location: lumbar, intestinal, intercostal, and such. From the diagram, we see that a number of lymphatic vessels travel collectively through sections such as the lumbar trunk, intestinal trunk, and ultimately to the thoracic duct, being the largest of these lymphatic vessels, leading back into circulation. Accordingly to the way that the collecting ducts are arranged, if you can see in this picture here, the area shaded in red would be the area serviced by the right lymphatic duct, draining lymph from the upper side of the body, whereas the remaining portion of the lymph is collected via the thoracic duct and other vessels. Much like we saw with the circulatory system last week, the direction of flow, like we see through some of the arteries, arterioles, and capillaries and veins in the circulatory system, begin with the lymphatic capillary. Remember, that's a terminal end pathway surrounding some of the circulatory capillaries, then in through a lymphatic vessel, which contains some of those flap-like tissues of valves. Managing through a lymph node, we'll be looking at lymph node structure shortly, into a larger lymphatic vessel, ultimately leading to some of the trunk pathways, the collecting duct, and then ultimately back into the subclavian vein for return of that fluid to the circulatory system. We mentioned that this lymph is basically this plasma-like solution that contains mostly water, some dissolved minerals and such, very small proteins, and is formed from the pressure exerted on the walls of capillaries with this interstitial fluid being picked up by these lymphatic capillaries. The formation of that lymph, though, is dependent on tissue fluid, this where it's derived from. This tissue fluid we know comes from blood plasma, and so any items that might be found within blood plasma, such as water and some of those dissolved substances, would be found in lymph. But remember that endothelial tissue can act as a membrane, really filtering out some of the larger proteins. What happens, though, it does contain smaller proteins. Proteins that are less than 30,000 molecular weight, which is just a unit, very small. And what happens is this does change the osmotic pressure. We know, again from last week, that osmotic pressure is derived from the ability of a fluid to draw water if it has a higher solute concentration, like those proteins. Now, because of the nature of those lymphatic capillaries, allowing a certain amount of material and water through, this increases the interstitial fluid, which increases this hydrostatic pressure within the interstitial spaces. This is going to aid the movement into lymphatic capillaries. In addition, there's these sort of micro flaps of tissue on the membrane itself that, again, act like a valve to encourage the one-way movement of this fluid into a lymphatic vessel, and then we can refer to it as lymph. Now, if this process is inhibited in any way, can be due to a variety of reasons, we're not going to have or encourage that positive movement of fluid into the lymphatic vessels, and we can end up with a situation referred to as edema. Now, last week we had, or previously in the week before, mentioned the role of fats. Fats, we know, dietary fats are used in metabolism, and there's a process of reformation of these fats in order to transport them throughout to our tissues. We start with these chylomicrons, which are made from micelles, and these chylomicrons are transported to the liver, where they're conformed into VLDLs, very low density lipoproteins. These are then converted into LDLs. As you can see, LDLs transport cholesterol to body cells. Now, I've written here bad oftentimes when our cholesterol is checked on the amount of transport of triglycerides to our cells versus the HDL, which is the return of cholesterol back to the liver, is out of balance. And we can end up with a concentration of cholesterol building up on the walls of blood vessels, and obviously that would cause some atherosclerosis and some other problems. But the point here is that these fats, these triglycerides, are transported via these protein molecules and delivers fats to the bloodstream, where they're delivered to cells to be used during metabolism. In addition, we mentioned a few of the other functions of lymph, moving tissue fluid to the bloodstream, delivering other particles to lymph nodes to be filtered and such. We see here, if you remember, the pressure differences between the endothelial lining of the lymphatic vessel and the capillary, and the interstitial fluid is very low. So we see here the filaments anchored to the connective tissue really aid in the positive collection of this lymph, and then movement of lymph out of the vessel back towards the circulatory system. Now, because the pressure is so low in that interstitial exchange, we really rely on a few other mechanisms other than hydrostatic pressure to move this lymph along the capillaries. For example, the action of skeletal muscles, the actual contraction force with these muscles riding alongside of these vessels. Respiratory movements, that change in intrathoracic pressure that we had discussed previously. Smooth muscles in larger lymphatic vessels, that they may aid that, and also those flaps of endothelial tissue referred to as a valve. All of these are going to help maintain that positive movement of lymph. Eventually, depending on the pathway, these lymphatic vessels here you see they're referred to as afferent. Afferent just means a direction. It's bringing this lymph to these lymph nodes. And you see the efferent vessel here means bringing this lymphatic fluid that's been filtered by the lymph node away. Within the lymph node, we see a number of interesting things. First off, macrophages and T cells, and some of those B cells we said that are sitting in this germinal center, this area where these cells congregate. But around the area is a sinus. A sinus is an opening where this fluid can float and move across these germinal centers and literally be monitored for the presence of anything foreign that shouldn't be there. And obviously the white blood cells would be right there to respond to anything that might be present. The exit portion of the lymph node is referred to the ileum. Again, a lot of these structures you will see again the nomenclature when we study the kidney. There's some blood vessels that are bringing blood to and from the lymph node, and lastly that efferent vessel. Notice the flaps again; those valves that are encouraging that positive movement through the lymph node. Now these lymph nodes that are placed throughout the system, a collection of them we call a plexus, and so you have a cervical region or a cervical plexus, an axillary plexus, an inguinal plexus, and so forth. Collections of these lymph nodes throughout the tissues. Here we see some functions of lymph nodes. We've mentioned the filtering the harmful particles from the lymph. Here it says immune surveillance by macrophages, which typically would be things like monocytes that we refer to them. They have a different name, same type of white blood cell, but when they leave a blood vessel, we refer to them differently. And also those lymphocytes, those T cells and B cells that are monitoring our immune system. Here we see the thymus gland. Thymus gland sits superior to the heart. It's bilobed. It's parasagittal to the trachea. It says it's typically small in an adult. Site of T lymphocyte production. This is where those T lymphocytes mature. It says they secrete thymosins, which are hormones that really guide T cell lymphocyte production. Spleen is another organ of the lymphatic system. Look in the upper left abdominal quadrant. Very vascular. You can see the dark colored tissue with two centers, white pulp and red pulp. Now we had seen under the microscope last week in lab where these tissues appear different colors. This white pulp is actually white or whitish looking in the microscope. It's where we have a high concentration of those T cells and B cells, whereas red pulp obviously contains a greater portion of blood cells in addition to some other white blood cells. Okay, question slide here, so why don't we take a short break. Please go ahead and answer those few questions and I'll come back and we'll give you some response. As we move away from anatomy, we're now going to move into a section on physiology, looking at the lymphatic system, how it in fact can help us fight or defend ourselves against foreign agents we refer to as pathogens that can cause infection. Pathogen is referred to as a disease-causing agent. There's many different types of organisms that can do this, as you see here: bacteria, viruses, spores, other protists, like this is when they say complex microorganisms, and sometimes even proteins can be considered, mutant proteins called prions. Can be considered pathogens. Our body responds to those in a two-step process. We refer to these as either innate defenses or adaptive defenses. Oftentimes these innate defenses would be sometimes referred to as non-specific or cellular-mediated immunity, CMI, whereas adaptive defenses, other terms you may hear, would be specific defenses or antibody-mediated immunity, referred to as AMI. There are a number of non-specific or innate defenses that we see with our immune system. If you look at this list, many of them make rather sense. Species resistance. We're based on our unique genome. We only respond to certain diseases that affect our particular species. Mechanical barriers, like the skin. Chemical barriers, like our stomach acids, like the natural antibiotic in our eyes. Something referred to as NK cells, or natural killer cells. We'll talk about this a little bit later. And also this process of inflammation, which we will mention in a moment. Things like fever and phagocytosis. Again, all of these respond non-specifically. It doesn't matter what the type of organism it is. There's a defense that's been activated, and all of these will help to inhibit the future growth and spread of this pathogen in our system. The mechanism for this innate system basically is four processes that will happen, again, regardless of the specific type of infectious agent. There's something called opsonization, enhancing the phagocytosis of antigens, basically recognizing and being able to engulf anything that's foreign. We've talked about this one, chemotaxis. There are chemicals that help attract those macrophages, those monocytes and neutrophils, to wherever that site of infection may be. There's a rather interesting system, this process of rupturing foreign cells. If in fact a foreign agent is cellular in nature, as you can see, there's a series of proteins that will embed themselves in the cell membrane, basically punching holes in the offending organism's membranes. Excuse me, I lost my voice there for a second. And basically, if you rupture the membrane, that's referred to as cell lysis, and the cell cannot survive. Last one, agglutination. We've taken a look at this one when we studied the ABO blood system and how that process can be triggered, this process of agglutination. Now, one of the last innate responses we mentioned on the previous slide was inflammation. Inflammation is a normal tissue response to injury. As you can see here from this slide, there's a number of things that will take place when there's been damage to cells. If you remember, there's a number of chemicals that are found within cells that normally should only be found within their cytoplasm. There's a group of molecules referred to as interleukins. If those are detected outside the bloods of normal body cells, that signals an injury response. And the tissue response would be things like dilation of blood vessels, capillary permeability increases, phagocytes become active, tissue fluids containing clotting factors seem to the area we want to obviously stop any blood flow in that area before we get to any kind of repair process. So regarding the last set of slides, really sums up the innate or non-specific defenses. These defenses are activated the same way and processed independent of any type of particular pathogen. If our innate defenses fail, our first lines of defense, if you will, our body will initiate specific defenses, or what we refer to as adaptive portion of our immune response. These are resistance to particular pathogens. Depending on if you are a bacteria, a virus, or a mutant protein. The ability for us to do this, remember, comes from those markers, those surface antigens that helps us detect self from non-self. And the presence of a non-self antigen obviously will elicit some kind of immune response. Just a quick slide on those surface markers, those antigens. We know that they are proteins. They can be a variety of other, or composed of a variety of other macromolecules. They are fairly large proteins, and again, help determine self from non-self. Now, for a cell to determine or to make that determination of self from non-self, there's a process of maturation that needs to occur, specifically in those T lymphocytes. As we can see here, we start at the number one prompt. Stem cells in red bone marrow produce those lymphocyte precursors. They're not effective yet. Just like we see with young men and women, we often send them to a training boot camp, we call it, for the Army and other armed services, where they learn to fight the enemy, who the enemy is, but that takes a great amount of time. Same thing here, these lymphocyte precursors, they're marking these as, they're noting these as B cells and T cells. They need to become further matured. Once those T cells have spent some time in the thymus, again, I've mentioned this before, but there's a protein called MHC, major histocompatibility complex. Basically, that's the protein that guides this maturation, the determination of these T cells, so they can identify any foreign antigen. It gives us that ability to then respond to those foreign antigens. And once that happens, both B cells and T cells are transported through the blood to various lymphatic organs, like the spleen, or here we see a lymph node. Now on this slide we see the variety of functions of T cells, and oftentimes I will group T cells into two main groups. You can see here there's a subset. Some T cells we refer to as helper T cells. They help activate other T cells and other cytotoxic T cells we see down here. Oftentimes they're named for the proteins that they contain. For example, the helper T cells contain the CD4 protein, whereas those cytotoxic T cells, or referred to as killer T cells, contain the CD8 protein. It's just a way to identify them. These T cells, specifically helper T cells, says they activate cytotoxic T cells, killer T cells. They also will stimulate other cells like the B cells to mature and develop into something referred to as a plasma cell, which we know produces antibodies. And antibodies are very powerful proteins to help defend our system. A couple of other subset of function here. They says they secrete growth inhibiting factors. Again, that's going to make it very uncomfortable for this offending pathogen to proliferate. And secrete interferon. Again, the same thing. Interferon to interfere with this disease process of this pathogen. B cells, however, really don't interact directly with the pathogen, but they produce something very powerful called an antibody. They differentiate into plasma cells, it says here. We had mentioned some different types of response. They refer to this as humoral immune response, or sometimes I refer to this as AMI, antibody-mediated immunity. They're going to produce those antibodies, much like we saw with blood typing. Those antibodies, those Y-shaped proteins, we'll take a look at their shape in just a moment, really are produced in high rate and then can act upon the foreign pathogen. So from T cell and cellular immune response, we said that previously this T cell needs to bind to a foreign antigen. Okay, so that foreign antigen, whichever particular antigen is present on a particular pathogen, immediately sets off a cascading series of reactions. The ability to detect those foreign antigens comes early on in the life cycle of those T cells from the MHC, and the MHC guides the formation of very specific T cells, each one having a unique recognition antigen to detect those particular proteins on the pathogens. I had mentioned two types of T cells previously, helper T cells, the CD4, and the cytotoxic T cells, the CD8. One I did not mention was the memory cells. We'll see in a moment what those are for, because we can really look into the idea of a primary versus a secondary immune response. Obviously, a secondary immune response is going to end up being much more rapid than a primary response. So how do these T cells, in the presence of an antigen, activate and turn on our immune response in this process? So we begin looking at these this particular cell. This is a macrophage, and as you can see here, what it's done is it's basically phagocytized a pathogen, in this case probably a bacteria. On the surface of the bacteria you have antigens, and it has now digested that bacteria and displayed these antigens, cut it up into small pieces, and displayed these antigens on its surface of its cell membrane. Now eventually, a helper T cell will bump into one of those and recognize that very particular antigen. It contains a very particularly shaped receptor. And once that receptor antigen complex forms, a trigger is switched. And that T cell immediately will start to interact with other cells and release something called cytokines. Here it is right here. Cytokines are chemicals that are going to have a variety of effects. One of those effects is to trigger mitosis, which increases what they call the selection of T cells with that very particular protein. In addition, cytokines will help stimulate the correct B cells to proliferate. They'll also increase mitosis in B cells. Now, those B cells have also a very particular shaped protein called an antibody, and it will start to produce those antibodies, and they can bind to those foreign cells and cause some inhibition. So the next step in this process, we need to form an increased number of antibodies. So we can see that antigen that's been present on that foreign cell has bind to an antigen receptor. In addition, alongside with the help of a T cell releasing cytokines, we see these B cell proliferation, which basically means a cloning, increasing mitosis of that very particular antigen, and we can increase the number of these cells. All of this process, though, does take a certain amount of time, and that's the problem. Oftentimes pathogens can cause or initiate a disease process much faster initially than we can respond to them. Given enough time in this process, we see that the B cells previously from the previous slide, the activated B cells that have been stimulated via the receptor antigen combination or the presence of those cytokines from the T cells causes proliferation. We have our clonal selection of B cells. Now those B cells could do have a few pathways here. Number one, they can differentiate into what we refer to as plasma cells, and where we can increase the production of antibodies. I believe upwards of 2,000 antibodies per second per B cell can be produced. So we saturate our bloodstream with these antibodies designed for that particular pathogen. In addition, we form memory cells, which are those B cells that already have the very particularly shaped surface antigen, and it can produce the correct antibodies in a much higher concentration than we originally started with. This is a nice summary slide of the steps that we had just talked about in formation of antibodies. So we see in this slide the structure of an antibody molecule. It is the end portion, the epitopes, if you will, or antigen binding sites that really provide this molecule with its unique ability to interrupt the cycle of infection for some of these pathogens. Each antigen binding site is very unique. Okay, and it's only the particular B cell that has been activated that's going to produce this monoclonal selection of antibodies, which again does take some time, but a very powerful process. As we discussed previously, the initiation of the immune response, the activation of the helper T cells, killer T cells, and plasma B cells really revolves around the initiation of that process, the increased mitosis of those specific cells, B cells and T cells, formation of those proteins. All of this does take a bit of time. We can see here a primary response, the first time that our immune system has been triggered for this specific pathogen, can take a number of days after exposure to develop a certain plasma antibody concentration here, enough to significantly interrupt the infection process. Here, on the secondary response, we note that there's a much rapid production of antibodies after exposure to that particular antigen. We call that a secondary response. Now there's a number of different factors that can determine a secondary response. Subsequent exposure to the infectious agent, things like vaccines, and such. In addition to the lesson this week, I've also posted a discussion board this week. If you could log into Blackboard and take a look at that assignment. I would like you to investigate this process of vaccination. Now there's been some mention in the news most recently of individuals that are choosing not to vaccinate due to a number of reasons and factors. It has to do with this idea of primary or eliciting a secondary response. So once you've done some research, go ahead and log into the discussion board, complete that assignment. I would suggest doing one fairly early in the week. And then also I'd like you to respond to at least one other comment that individuals are making. Again, remember to let's base this on cited research. We see a number of different classifications of immunity. The first one, naturally acquired active immunity, basically exposure to a live pathogen. Next, artificially acquired active immunity. This would be something like a vaccine, where we have developed a vaccine that contains either a weakened, it's referred to as an attenuated virus. Sometimes it's simply the virus, it's the components that are extracted and then administered. Oftentimes we see some general types of malaise as a result of these injections, but usually that's a function of our immune response that's producing those symptoms, because you're actually reacting to the presence of that disease pathogen without it having the disease process. A few others, artificially acquired passive immunity. Sometimes we can subsequently aid someone that maybe cannot form an immune response, someone that's immunocompromised or whatnot, by giving them gamma globulins injections that will help temporarily improve their response to that pathogen. Or lastly, naturally acquired passive immunity. We see this during early development through breast milk. Again, where the recipient is not able to mount an immune response, so we're providing some temporary source of those proteins. Now, interestingly, our immune response can get out of hand if our body or our immune system reacts to something typically that's non-pathogenic. We refer to as this an allergic reaction. The reason why this can be dangerous is it can, like it says here, damage our tissues. Beginning with that initial contact, much like we would see with any normal immune response, our lymphatic system, specifically here a B cell, is combining with what they refer to as an allergen. It's non-harmful, but it's going to elicit the same response, immune response that we normally see if it was disease-causing. Those B cells will differentiate into plasma cells, releasing the IgG antibodies. Those antibodies bind to a particular receptor. Here's where the problem comes in on something called a mast cell. Now, a mast cell typically is a basophil that has left the blood vessel. It is now out in the tissue, and mast cells, or basophils, as we remember, have an ability to release histamines. Now, histamines promote inflammation. These are chemicals that can trigger massive amounts of inflammation. Basically, our capillaries become more permeable to fluid and proteins. The fluid in the interstitial spaces begins to build up, and we know that as tissue swelling, it can cause a variety of symptoms, typically that we refer to as an allergic response. These allergic responses can be broken down into 4 types, if you will, depending on the symptomology and causes for the allergic reaction. Type 1 would be sort of an immediate reaction, as it says here. And they show you some examples occurring within minutes after contact with the allergen. One of the more dangerous results of a type 1, because it is so immediate, is that interstitial fluid movement from the plasma into interstitial fluid can happen so rapidly that our overall pressures will begin to drop, and we refer to this anaphylaxis or anaphylactic shock. It is a sudden loss of blood pressure due to all that fluid becoming interstitial at a rapid pace. A few other allergic reactions that we might categorize here would be type 2, sometimes referred to as a transfusion reaction. We might see if there'd be an incompatibility issue with a particular blood type or something. Can take a little bit longer to develop. Or type 3, which would be obviously associated with damage of body tissues. The last one being categorized as a type 4 allergic reaction, with a delay a little bit longer to initiate immune response. Usually exposure to allergens superficially can produce a variety of results. For For those of us that work outdoors, sometimes things like contact dermatitis and poison ivy and things like that can be a real problem. I've exhibited that many times. So many of us are very familiar with a type 4 allergic reaction response. Because of the nature of our immune response, really detecting any foreign tissue or cell or protein, this also initiates an issue with transplanted tissue. We previously looked at blood typing and the ABO blood groups, and really we were circumventing, when you identify someone's blood type and then administer some blood products, you're circumventing the body's immune response. The body does not see that as foreign, as self, and will accompany that. Anytime we have transplanted tissues and organs, obviously those have also been marked with certain surface markers developed by the MHC. And we have the idea of rejection. The tissue rejection, very similar to how we respond with CMI, cellular immunity, against antigens. We can offset that to a certain extent using immunosuppressive drugs, which would, basically inhibit our immune response to a certain extent. Now, because of this tissue rejection process with a number of organs and tissues that can be grafted, some better than others, as we see here, an isograft, which would be either a donor would be an identical twin, for example, bone marrow, they mentioned here, or an autograft. Both of those would produce very favorable types of grafts where the grafted tissue would be incorporated. There would be no response because it has those particular markers that cannot be determined as non-self. Although you can see a few examples here. An allograft would be the same species. They mentioned a kidney transplant from a relative or a closely matched donor. But again, a compatible donor needs to be a match for greater than 6 alleles. I believe there's 13 that they test for, and you have to be a match for greater than 6. So it's not a perfect match, and allografts can be successful, but again, a regimen of those immunosuppressive drugs need to be administered. Sometimes we can use other tissues, a xenograft from other species like heart valves from pig and other such things. But again, the rejection can be an issue. Another issue with our immune response that can become a problem is this idea of autoimmunity or autoimmune disorders, where the ability, as it would make sense, an ability to distinguish self from non-self. There's a number of different disorders you can read about here. Interestingly, a few examples: type 1 diabetes. There's some research to suggest that those beta cells in the pancreas that stop producing insulin are attacked by our own tissues. Lupus, ulcerative colitis, RA, rheumatoid arthritis, another one that is fairly common. Those are cells lining the joints. A number of different disorders where the body's lost that ability to make that correct identification. An interesting note here, we know that because of the anatomy of our immune system, the thymus gland changing size, our immune response is a slow and steady progression. We're at higher risks of infection. Our response to producing certain antibodies and to effectively against those foreign antigens becomes slower. The concentrations of antibodies decreases. All these together really dampen our immune response as we get older. One final response to normal function of our immune system would be HIV. This is a virus that causes AIDS, and I have written here immunity breakdown, which basically those cells that are really important for initiating our immune response, those helper T cells, the CD4 cells, are attacked by HIV, the HIV virus. Okay, we know that AIDS basically is a slow progression or weakening of our immune response. HIV, it says, impairs macrophages and helper T cells. Now, the helper T cells, those are the cells that initiate our immune response. These helper T cells that contain the CD4 protein, initiate our immune response. The HIV virus contains a surface protein called GP120. GP120 is what the CD4 will bind to, and that allows an entrance into the white blood cell of this virus and containing the viral genome. And once that viral genome enters the T cell, it attacks the T cells and will ultimately impair their function. As far as a normal response, HIV still is a virus, and our body does have natural ability to detect, foreign proteins, foreign virus particles. The problem that HIV presents, though, is it does mutate quickly, so that as our body, our immune system, is trying to develop an innate response, HIV can change and mutate. It's been said that some AIDS patients can have upwards of 100 different strains of HIV virus within one individual. So as you can see, it provides a really difficult situation for our immune system to try to keep up with each individual strain. The end result is a significant decrease in the concentration of those helper T cells. Now, those helper T cells are the cells that initiate the immune response. So someone with HIV, as we see above, that produces AIDS, suffers from weight loss, fevers, weaknesses. They really have virtually no immune system to respond to, and oftentimes AIDS patients will typically die from things like pneumonia, certain types of cancers, and other general diseases that with a healthy, normally functioning system would be no problem. But because they lack the concentration of those helper T cells, that problem becomes even more significant. I hope you've enjoyed this rather brief introduction to the lymphatic system. I would suggest now moving on to your weekly worksheet, which is posted. Also, some research as far as the discussion board, to do some research and then respond to that prompt. And then when you're finished with both of those assignments, go ahead and move on to the quiz. I hope you guys have a great week, and we'll see you next Thursday. Don't forget we have an exam coming up, so study hard. Download those study guides. Have a great day.